Thank you, Uli. Thank you, Jim. And thank you to the other organizers who've invited me to share a bit about our research. And so I'll be talking about, again, another system which may or may not be by itself prebiotically plausible or relevant, but could inspire other types of systems that could be themselves relevant. So just to start, so a lot of us in biology or biochemistry, we've been studying how the cell works, and the cell is the basic component of life consisting of a number of very complex structures and chemical reactions. And we've come a long way to understanding how the cell works, yet there are still some unanswered questions. And one of these is, what exactly did a primitive cell look like? How did the first cells on Earth come to be in the first place? And of course, as we've heard throughout the day, there are some ideas about what this primitive compartment may have looked like. For example, perhaps some type of genetic or metabolic materials inside of a membrane-bound structure. And so one of the things that I'm really interested in is to see how we can build this type of cell, this primitive compartment-like system, from available prebiotic chemistries, and eventually observe whether these primitive compartments can eventually evolve into structures that may have resembled modern cells. And as I don't need to state this further, but compartments themselves may have been very crucial to the origins of life, as various compartments can perform many essential functions such as concentrating and segregating reactants, protecting molecules from degradation, and many other processes. And one of the ways that compartments could be generated, as Kepa graciously was able to introduce, is through a process called liquid-liquid phase separation. So this is a process that occurs throughout biology in the modern day. In our cells, we have many phase-separated organelles, and this process results in, for example, the formation of a membraneless compartment that can compartmentalize molecular components and also allow exchange of molecules between phases. So some of these examples in modern biology are stress granules, or there's even some suggestion that chromatin contains some of these membraneless organelle-like domains. So this phase separation process may have been one mechanism by which a primitive compartment could have assembled. And these phase separated structures, they could form in a couple of different ways. One on the left is through a non-associative interaction, and the one on the right is through something called an associative reaction. And so, for example, on the left, non-associative, this mode results from, for example, when there's two polymer components that don't like to be together due to differences in chemistries or differences in state. And so this results in formation of these droplets, which is exclusionary of one type of component, but inclusionary of the other type of component. Whereas in an associative type of phase separation, this occurs when you have, for example, oppositely charged polymers like nucleic acid and a positively charged peptide. These bind together electrostatically and through other interactions, forming these nice membrane-bound structures that are inclusionary of both types of polymers. And I'll say here, for purposes of simplicity, I'll refer to coacervates as two-component systems. You can create these coacervates or associative phase-separated structures through one or more than two types of components, but for the simplicity of this today's discussion, I'll refer to those as simply two-component systems. And so these coacervates are actually used as membraneless compartment models in various origins of life related research investigations. And they actually perform a number of interesting functions that may have been relevant for primitive life. For example, it's been shown that membraneless compartments can segregate and concentrate biomolecules like magnesium, which we've heard a lot about today, nucleotides, or even nucleic acid oligomers. Some of these coacervates can actually grow and divide through the exertion of outside forces, like through electric fields. And it is possible to have coacervates with multiple phases, such as those seen on the left and the right. And these phases might have different chemical affinities or provide different types of chemical media for different reactions to occur. And this allows these structures to actually increase its complexity, and further increases in complexity could be could emerge through, for example, scaffolding of membranes. Here we see that membraneless droplets can actually scaffold the assembly of a lipid layer around itself. So perhaps leading to a transition from a membraneless to a membrane-bound primitive compartment type of structure. And so one of the things that I, and along with my collaborators and I have been interested in is how to increase the structural complexity further of these coacervates, as one could imagine increases in structural complexity could lead to further increases in functional complexity. And so one of the structures that that my collaborator Tommaso Fraccia at ESPCI in Paris has been working on for a long time are DNA liquid crystals. So these liquid crystals are formed from nucleic acid oligomers. They're very short oligomers. This on the bottom left, this is one example, the specific one we used. This palindromic oligomer forms these short DNA duplexes, which aggregate end to end through their blunt ends, and they form these long stiff rods. And these rods, at sufficient high concentration of DNA, for example, through drying, can form liquid crystal phases through the bundling of multiple rods together. And as you see here on the right, this is a polarization microscopy image of a liquid crystal formed just through DNA. So we were interested in seeing whether we could incorporate this liquid type crystal type of structure into, for example, a coacervate type droplet, which was composed of DNA. And in this case, we chose a cationic peptide polylysine, as DNA and polylysine has been shown to form coacervate droplets in a number of conditions. And we believe that, although we didn't show this in this study, we believe that, for example, introducing liquid crystals into the system could help the system to incorporate some interesting properties of liquid crystals, such as supramolecular chirality, or perhaps even selectivity of certain chemical reactions down the road. So first of all, we tested a bunch of conditions. We tested many conditions combining these DNA duplexes with polylysine. This is fairly long polydispersed polylysine. And we found that we were able to find a condition where the liquid crystal structure and the coacervate droplet co-localize. So on the top is a standard bright field microscope image. On the bottom is a polarization microscope image showing this typical fingerprint structure of a liquid crystal. So we found these conditions, and we noticed that these conditions are fairly narrow. So here we did a salt titration, sodium chloride titration, and we noticed that the co-localization or the assembly of a liquid crystal coacervate only occurred at very narrow range of salinities. And we believe that this is because at lower salinities, as you see on the left, it's likely that the DNA and the peptide are interacting much more strongly, resulting in, for example, solid like precipitate formation. Whereas at higher salinities, we can see that at first the liquid crystal phase disappears, and then the entire complex actually disassembles as well, likely due to the disruption of these electrostatic interactions between the DNA and the peptide. We also went on to test whether this end-to-end DNA interaction was in fact very essential to the formation of these liquid crystal coacervate droplets. So we did the same experiment, the same tests with a DNA, palindromic DNA duplex, instead of with a blunt end, with a 2 T base overhang. And we observed that in theory, this overhang would prevent blunt end stacking. And so we observed that although we could see the formation of the coacervate itself, in no conditions did we see the liquid crystal formation, suggesting that this blunt end stacking is absolutely essential to the liquid crystal formation process. So now we have some idea about the conditions in which these structures can form. We next wanted to test whether these structures could, what their reaction to various perturbations was. So the first thing we tested was dehydration. And so we took some sample and we simply dried it. And over a period of minutes and many minutes and up to an hour or a few hours, we noticed that the structures actually were able to transition and eventually disassemble. So for example, starting from this liquid crystal coacervate state, after drying, the liquid crystal structures actually disappeared, followed by disappearance of the coacervate droplets at large. And we believe that this may be due to up concentration of the salts already in the mixture. And so, dehydration might up concentrate these salts, resulting in a similar transition as we observed when we did a simple salt titration. Next, we're interested in studying the reaction to temperature changes of these liquid crystal coacervates. And so we started actually with a system of a precipitate-like structure of DNA and peptide. And through heating the structures, we noticed that at around 60 degrees, these precipitate-like structures eventually transitioned into perhaps a liquid crystal coacervate-like co-localized state, followed by, at higher temperatures, the existence of a pure non-liquid crystal coacervate. And then upon decreasing temperature, we noticed that eventually some of these liquid crystal states emerged. And finally, at very low temperatures, the liquid crystal coacervate droplets actually became the preferred structure instead of these precipitate-like structures. So we believe that perhaps this type of heating step, this annealing cycle, if you will, might be necessary for these structures to find its local minimum, or the global minimum, where the structures exist in their most stable state. And so here's just a little bit of a summary. We actually were able to see so B on the bottom, this is the structures after heating and cooling, and we see the formation of a few different types of liquid crystal structures co-localized to the coacervate droplets. Don't worry so much about the notation, but these are all typically found columnar or other types of liquid crystal structures found in non-coacervate liquid crystal structures. And so with that, here's a little bit of a summary, and this is what we believe might be going on inside the droplets. So at very low heats and salinities, we have this tightly packed columnar liquid crystal state co-localized with these droplets. And this is where these long stiff rods in blue of DNA interact with peptides in red. These long stiff rods will bundle together into hexagonal patterns. And upon increasing heat and salinity, these rods will start to slowly disassemble, forming a different type of liquid crystal structure. At even higher temperature or salinity, for example, these rods start to unstack, resulting in disassembly of the liquid crystal structure, but still retention of the droplet structure at large. And finally, at high enough heat or salinity, all of these interactions are inhibited, resulting in disassembly of the entire structure. So if you're interested in hearing a little bit more about the experimental details, please feel free to check out this paper that we wrote last year, or another one which is a little bit more less physicsy and more related to origins of life, prebiotic chemistry. And I'll say that we're really interested also in, I'm really interested in studying other types of primitive membraneless compartments. So here's we've also done with Jim Cleaves, who's here, and with some others, we've done some work with droplets made of polyesters, which were synthesized through dehydration synthesis of alpha hydroxy acid monomers. So if you're interested in learning more about these, you can check out some of these other studies that we've published as well. And so I'd like to thank my collaborator on the DNA liquid crystal project, Tommaso Fraccia, and also these great grants, especially the French Embassy in Japan, who supported this project through a collaborative research travel grant for me to go visit Tommaso's lab in Paris. And we did a lot of experiments over the time. I'd like to thank my other collaborators who have been working with me for many years on the polyesters project, and also other members of ELSI and Tokyo Tech, where I'm based, for their contributions to these and other projects as well. And so finally, thank you for giving me this opportunity to speak. If you want to contact me later, please feel free to contact me by email. And after this session, if you want to learn more about ELSI, there's an ELSI table in the, I guess it's called the My the Meet Anyway platform. So please feel free to join the LC table if you want to learn more about education, research, or job opportunities at LC. Thank you very much for your time and attention. I'm happy to take any questions.